Thank you.
It's certainly my pleasure to introduce Marylyn Ritchie, who has been now at Penn State University for a while. She's director of the Center for Systems Genomics, and is going to talk about using GWAS and prior knowledge to uncover gene-gene interactions. Marylyn?
I'm here. Thank you very much, Nancy. Can everyone hear me okay?
Yes, you're very clear.
Okay, great. Well, thank you for this opportunity to speak as part of this webinar today. I really appreciate the invitation. And today I'm going to talk about how we've been using the GWAS Catalog and information in the GWAS Catalog as a way to look for gene-gene interactions. And as a bit of introduction, I thought it would only be right to show a picture of the GWAS Catalog at a webinar like this. So as we know, the NHGRI has done an excellent job of curating all of the Genome-wide association study hits that have been found for many different traits and diseases. And as of earlier this month, there are over 10,000, close to 11,000 SNPs, and probably at this point in the month, it is over 11,000. And there are over 1,600 publications. And so genome-wide association studies have really taken off over the last 5 years and in many ways dominated the literature in our field. And so this has been very exciting, and we know that we now have a lot more knowledge about SNPs associating with complex traits. But one of the things that we routinely see is that the effect sizes or estimates of heritability explained by these hits is generally very small. And the slide that I'm showing now is actually a slide from Terry Manolio from several years ago, around the time that they published the PNAS paper. And I haven't updated the data in the slide primarily because I believe that what we're seeing in the lower left, the low odds ratios, those bars would only go much, much higher. Because as we've seen, the number of associations that have small effects continue to be very small. And as we've increased sample sizes and started to build consortia to join data sets together, it's enabled us to find even more genome-wide significant SNPs. However, many of those have been even smaller because we needed a larger sample size to find them. And so we have very few associations with odds ratios that are very high, out in the 3, 4, 5 range. And many of those in that range we actually knew before genome-wide association studies, and in a lot of ways those were our proof of concept hits that kind of validated that the GWAS approach worked. And so a lot of people have been talking about the fact that we're finding very small effects. There are some papers that refer to this as the low-hanging fruit, where the predominant model that we've looked at are just single additive linear effects, just one SNP at a time, and what we're picking up are the low-hanging fruit in the trees of genetic architecture, and that there are actually a lot of nonlinear effects that are there to be found, and we just haven't found them yet. Others talk about it as the case of the missing heritability, that there are a lot of reasons why the genome-wide association study hits don't explain a lot of the heritability of these complex traits. And this is a very popular paper by Brendan Mayer that describes a lot of the areas where this heritability might be. And while we're working on a number of these, the out-of-sight idea is that of rare variants, and many of us are working in that area. In the architecture is referring to structural variation or epigenetic variation, so copy number variants or other structural variants. And the one that I'm going to talk about today are the underground networks. And this is the basic idea that we have gene-gene interactions or pathway or network effects that are truly important for complex traits. And by looking one SNP at a time, we're missing some of the heritability because it's primarily through its interactions with other genes or proteins that these effects can really be recognized. One other thing I'll point out, the Great Beyond refers to gene-environment interactions, and some of the methods that I'll be talking about today we could use in the context of looking for gene-environment interactions as well if we expand the data sources that we're building to look for these gene-gene interactions. And the reason that I've been focused on gene-gene interactions for quite a long time, I'm a statistical and computational geneticist by training, but I primarily was a biologist before getting into computer science and statistics. And so I think a lot about the complexity of biology, and this is just one pathway out of the thousands of pathways that I could have showed. We see a lot of complexity in biological pathways where you can have a single gene, such as on the bottom right, this calcium modulator gene that can interact with entire pathways. So it can be upstream of a pathway and cause signaling cascades that can affect pathways. It can affect multiple pathways. It can affect multiple other genes. We have in the upper left of the pathway different feedback loops. It's well known in biology that we have compensatory mechanisms where there are different families of proteins or families of genes where if one is dysfunctional, another one can compensate. And so in my mind, in thinking about complex traits, the likelihood that a single variant in a single gene could lead to such a complex phenotype seems to have a low probability, and that it's more likely due to kind of a multiple hit hypothesis where you have variation in multiple genes that come together to lead to a complex phenotype. And one other thing that kind of definition type thing that I wanted to go through is the type of interaction that we're talking about. So epistasis or gene-gene interaction, epistasis is the technical term for non-additive gene-gene interactions, kind of in the absence of strong independent effects. And when I'm thinking about gene-gene interactions, I actually don't focus so much on whether it is only nonlinear interactions or including additive interactions or multiplicative interactions. To me, the primary goal is finding the set of genes that come together to explain complex trait heritability. And it matters less about the particulars of the mathematical formula or how those variables come together. But there is often a lot of confusion about whether we're talking about statistical interaction or biological interaction. And so what I'm showing here are kind of a figure to try to show both. So at the top, this is statistical interaction or statistical epistasis. And this is the concept that we're looking in a population of individuals for evidence that variables are interacting in a statistical manner to confer disease risk. And so this is evidence of interaction in perhaps if you think about a logistic regression model, that you could have main effect terms and interaction terms. It's saying that those interaction terms are adding above and beyond the main effect terms. This is in contrast to biological epistasis or biological interaction, which is what I'm trying to show in the bottom. And these figures are out of a bioessays paper from 2005 by Moore and Williams. But this is showing biological interaction in one individual. And so in an individual, we think about genes interacting in a way that perhaps a protein is binding to DNA. So the protein for which one gene encoded that protein may bind to another gene physically at the DNA level. Or that you may have protein-protein interactions, and you can think about the genes that encoded for those proteins as also interacting. So these are more kind of biological interactions. What we hope is happening in a data set, a population-level data set, is that if we find evidence of statistical interaction, it is indicative of underlying biological interactions that might be occurring. It may or may not be the case that that's always true. So we may find statistical interaction that does not directly correlate with biological interaction, and when that happens, those models are much harder to follow up and validate. But more often, if you find statistical interaction between genes that you can make a biological mechanistic argument that you could go into the lab and test, those are much easier to follow up. And so what I'll be talking about today is statistical epistasis or statistical interaction with the hope that models that we find may relate to underlying biological interaction. And so one thing that I want to talk about is why, kind of the motivation for why we're developing the approach that I'll talk about today. And that's if we have interactions that are important through their interaction effects and not necessarily all of the interacting genes having independent, statistically significant main effects, one thought is that we could exhaustively just look through all the genes. So if we have, 1 million SNPs captured from a genome-wide association study, can't we just exhaustively look through all the combinations to look for the evidence of interaction? And unfortunately, we can't do that, and it's purely computational. So if we had 500,000 SNPs, which are some of the earlier GWAS studies, we would have 5 times 10 to the 5th single SNP tests. So that would be just the 500,000 tests. There would be 1 times 10 to the 11th pairwise interactions. And if we think that we want to go beyond pairs, which I believe that we do for complex traits, it's likely not just a pair of SNPs, but probably a more complex combination of SNPs. That's a lot of interactions to look at. And so to put kind of computation times on that, to do the 5-way interactions, of which there are 2 times 10 to the 26th, if we use computers that could do 1 computation per second, it would take 10 to the 21st days to complete those 5-way interactions. And now our computers have gotten much faster in recent years, and our GWAS chips have gotten much bigger. So currently we often look at about 5 million SNPs. The pairs of SNPs would take about 144 days. Now, this can be circumvented by using a parallel supercomputer, and so we do this a lot, where we'll run it on 100 processors, and then we can actually get the pairs of SNPs done in about a day, which is not at all a problem. However, if we think about looking at even the 3-way combinations of SNPs, that's at an order of 10 to the 8th days. And so even with very large supercomputers, this can take hundreds of days to complete, or even thousands of days. And the 5 SNP combination models are still computationally impossible. And just as a frame of reference, because 10 to the 20th days is a lot, the Big Bang Theory was 10 to the 12th days ago. And so we're talking orders of magnitude beyond the length of time in order to compute these computations. And so we know that an exhaustive evaluation isn't possible, and so we have to do something smarter in order to look for gene-gene interactions in a genome-wide way. One approach that's being taken by us, as well as many others, is to look for interactions among the top hits from the single SNP analysis. So you can imagine doing your single SNP GWAS, creating a p-value threshold cutoff of maybe 10 to the minus 5 or 10 to the minus 4, and then looking for all SNP-SNP interactions in that top threshold of hits. And you can probably go to even 3 or 4-way interactions in that space, depending on the number of SNPs that you have. But the limitation there is that you would only be looking for interactions among things that have a strong independent effect. And it's possible that there are true signals that don't have an independent effect that would meet a p-value threshold. We know that that's true even from single SNP GWAS studies, that there are some true hits that are kind of down in that gray zone of a Manhattan plot. And so the approach that we've been taking is to use biological prior knowledge to look for interactions, and we're calling this a candidate epistasis approach. And so that's the approach that I'll talk about today. The tool that we've been using to do this is called the biofilter. This is a publicly available tool, and I'll show my web address at the end where this can be downloaded for use among individuals. And the goal is to integrate publicly available databases to try to build the relationships between gene products. And this builds on this idea of biological epistasis or biological interaction, where we want to look for evidence of statistical interaction among genes that we know to interact in a biological way. And the primary reason for that is something I mentioned earlier, this idea of trying to do validation or follow-up. We always look for replication of signals that we're finding, and that's kind of the first important step. But then beyond that, we want to be able to go back to bench science and validate things biologically. And if you find models statistically that really don't make a lot of biological sense, it's very difficult to go back into the lab and validate those. And so we've been taking the bias of looking for things that would make sense biologically and then seeing if we find association and evidence of interaction in our data. And this is a means toward integrating information from the genome and the transcriptome and the proteome into the analysis. Because as you'll see in a moment, the data sources that we're integrating are not just gene-related, but they actually cover some of the transcriptome and protein information as well. And so the underlying database that we have constructed is called LOCI, or the Library of Knowledge Integration. And this database is a SQL-Lite database that is integrating other publicly available databases. And so what I'm showing here are the different databases that we're using. So we're capturing information about genes and SNPs from dbSNP and NCBI, ENCODE. We're also looking at Ensemble currently to figure out how to incorporate the Ensemble gene IDs as well. We have gene and protein interaction information from things like BioGrid, MINT, and FormGKB, which includes a lot of pharmacogenomic-related gene, protein, drug interactions. We have pathway information from Gene Ontology, category Exome, and NetPath. We capture information about protein families, regulatory regions. We currently are using oregano, but we're currently looking into adding information from the ENCODE regulatory regions that have been learned over the last couple of years. Evolutionary conserved regions from UCSC, and then annotations from the NHGRI GWAS Catalog. And we're constantly expanding this database. One thing I would mention, if an individual wants to download this and use this tool, what we've written is a series of scripts that will go out and hit these public domain databases and extract from them the information that you would need to do a biofilter-based analysis. It does not download all of these other databases in its entirety, as that would be a lot of information. But this script will enable you to hit those databases and grab the information needed and then update at will. And so, these databases update their information on different schedules. Some are weekly, some are quarterly, some are biannually. And as a user, you can update your version of LOCI whenever you'd like by running the updater script. And so what you'll download from us is a set of scripts to build an internal LOCI database at your institution as opposed to downloading a very large database. This enables you to download things quickly because downloading the database in its entirety would take a lot of time, whereas running the script doesn't. And I would also mention, I think our internal LOCI database is about 10 gigs. And so we're not actually talking about a huge database, but certainly larger than you would want to try to transfer in its entirety. So this biofilter approach we've been using for a number of years. The first publication was in 2009. We've applied it to a number of different traits, and today I'll talk about two examples from multiple sclerosis and HDL cholesterol, and both of these have been presented at ASHG and then also published. And we're working on a number of other traits currently. And so the biofilter application can be used in a number of ways. One is a very simple GWAS annotation. And this is certainly a way that we are currently using the NHGRI GWAS Catalog. So if you run a single SNP association test in a GWAS, you'll get your RSIDs and your p-values. You can feed those into biofilter and then have them annotated based on what genes those SNPs are in, what pathways they're in, what biological interactions those genes are a part of, perhaps in protein-protein interactions. You can also find out what genome-wide association studies those hits have been seen in from the GWAS catalog. And I would also mention this can also be used if you're doing a phenome-wide association study, or PHEWAS. We've used this quite a bit in our phenome-wide association studies, where we'll take our PHEWAS hits, run them through the biofilter to annotate them based on what genes and pathways they're in, but also what GWAS studies they've already been associated with, which can become very useful when you're thinking about a PHEWAS and looking for pleiotropy. And so the biofilter can be used to automate this annotation process, annotating your SNPs based on all of this public information from these other databases. And then it can also be used to build interaction models. And I should also mention, it can also be used for filtering. So you can run your SNP list through Biofilter to get a set of SNPs to run if you only want to look at genes from certain pathways or certain protein families, et cetera. The primary goal of what I'll talk about today is using it for looking for interactions. So you can imagine doing a very conservative interaction analysis where you're looking for SNPs in genes that physically interact, where perhaps gene product 1 is in a chemical or physical reaction with gene product 2. And so you would take the SNPs from gene 1 and the SNPs from gene 2 and look for interactions or evidence of statistical interaction. You could also take a slightly wider net where you would say, let's map SNPs into pathways and look for interactions within pathways or interactions between pathways. And this is going to be a much higher number of tests, but significantly less tests than you would do if you were trying to do an exhaustive evaluation. And so this approach is the one that we've taken in most of our analyses and certainly what I'll be talking about today. So as with any approach, there are a set of pros and cons. If you run something like the biofilter, you have many fewer genes to explore, many fewer SNPs. It's fewer tests in terms of computation time and in terms of multiple testing correction. And the results can be interpreted in a pretty easy way. The downside, of course, is that we're limited by the current state of knowledge and the knowledge that actually exists in the databases that we're using. And we have a limitation of learning completely novel biology. So if there are biological relationships between pathways that are very, very disparate in terms of our current knowledge, we wouldn't evaluate those currently. And so that is a definite limitation. But you can learn new biology with respect to a trait. So when we set up these models to test, we are not considering the trait that we're looking at. So if you have an Illumina 1M data set and you want to map those SNPs to genes and then create the set of interaction models that those genes are participate in, that gene list and interaction model list would be the same whether you were looking at a lipid phenotype, a cancer phenotype, or a psychiatric trait phenotype. It's really specific to the SNP list that you start with. And so as I said, we've applied this to a number of studies. What I'm showing here is the publication on multiple sclerosis, and this is part of the International MS Genetics Consortium. In this study, we were looking at trios, and so these were 930 trios, about half from the U.S. and half from the U.K. This was Affymetrix 500K array data, so these were one of the first GWAS studies run on MS back in about 2005, 2006. And after QC, we did about 300,000 SNPs that we fed into the biofilter. We ran the biofilter and created a set of about 22 million SNP models to evaluate, and we used a conditional logistic regression analysis that would incorporate the family information. And so this is a very straightforward two SNP main effect terms and then the interaction model. And then we did a likelihood ratio test to determine whether the interaction was adding above and beyond the main effect. And what we found were two different interaction models, and I'm showing the pathway as opposed to the actual statistical models, but they're in the paper. So they're color-coded. The blue, the CHRM3 and MYLK, was one interaction model that we found not only in the IMSGC data set, but it replicated in two additional data sets. Or I should say it replicated and or confirmed. In some cases, we had an exact replication where the interaction term was statistically significant and the main effect terms were not. And in others, the main effect terms were also significant in addition to the interaction terms. And so one interaction model was the two genes that I just described, and then the other was between PLC beta 1 and beta 4. So these are the two genes in kind of a pink color. Out of the 22 million models that we found, these were the two gene-gene models that replicated, and we looked at thousands of pathways, but interestingly, both of these gene-gene models were in the same calcium signaling pathway. And so we thought that was very interesting and certainly provided evidence that the calcium signaling pathway was clearly associated with MS susceptibility in the 4 data sets that we were looking at. And the other example is from the eMERGE network. This is an NHGRI-funded network to look at electronic medical records in genomics. And this is actually from the phase 1 of eMERGE, where we had 5 sites across the U.S. This particular study was done with the Marshfield Clinic Biobank and the Vanderbilt Biobank. We genotyped all of the eMERGE sites on the Illumina 660 platform. And for this particular study, we were looking at median HDL. These were extracted out of the electronic health records for anyone having at least 2 HDL measurements in their record. These individuals were actually phenotyped and genotyped originally for other traits. So in the Marshfield Clinic, they were collected for cataract, and in Vanderbilt, they were collected having normal ECG measurements of the QRS duration. And so this is an excellent example of reuse of genotype data when you have electronic health records. You can pull a lot of other phenotypes out. And so this is the median HDL trait that we looked at. We had about 4,000 participants from Marshfield and almost 2,000 from Vanderbilt. And the way that we did the study, we used Marshfield as a discovery cohort and Vanderbilt as a replication cohort. We looked at about 22,000 interaction models because we required that the evidence of interaction be present in at least 4 out of the dozen databases that we had in the biofilter. And in the end, we found 2 SNP-SNP models, but actually 1 gene-gene model that replicated. So this is the same SNP with the same direction of effect in the 2 cohorts. And it turns out they were in genes known to be related with lipid metabolism. The interaction was between ABCA1 and LIPC. These both have been associated with LDL cholesterol and in some data sets, HDL cholesterol. And here we find evidence of interaction. Again, we used a linear regression approach to model these. So we had the 2 main effect terms and an interaction term. And the interaction between ABCA1 and LIPC added above and beyond the main effects and replicated in the 2 data sets. So to wrap up, I just wanted to talk about one other future direction that we're adding specifically for the GWAS catalog. You could think about having a pathway. And so in this particular example, there are a set of SNPs that you've genotyped from a particular pathway. Well, one of the things that we do is to take those SNPs and you could map them then to the GWAS catalog and say that you're looking at a particular trait, such as type 2 diabetes. You could look to see what are all of the SNPs that have been mapped associated with type 2 diabetes. You could then map those SNPs to genes and pathways using a biofilter. And then you could see what are all of the other SNPs that are linked with those SNPs in the biofilters from perhaps TAG and Reactome and NetPath. So these would be SNPs that we know or genes that are interacting with that original gene found in the type 2 diabetes GWAS in our LOCI database. And then we could use that kind of as a starting point to look for gene-gene interactions for type 2 diabetes. And we could do an exhaustive pairwise, 3-way, 4-way interaction search among that set of genes that, we start from a single SNP GWAS hit and then branch out based on biology to all of the other genes that are related to that original gene and then look for interactions among those. And so this is one of the applications that we're currently working on, kind of starting with that original GWAS catalog SNP and building out from there. So to summarize, Biofilter is a bioinformatics application that we've built to annotate, filter, and construct gene-gene models for evaluation. I didn't mention, but I probably should, that the Biofilter, as it stands, is decoupled from any particular statistical or modeling paradigm. So you could take the list of RSIDs that you get out of Biofilter and run them in Plink or in a multi-factor dimensionality reduction analysis or in a neural network analysis, or really any statistical or analytic paradigm. It just is constructing the set of models to look at. We've successfully applied Biofilter in a number of GWAS analyses to look for either replicating or confirmatory gene-gene models. In some cases, they replicated where, using the standard definition of replication, where it's the same SNPs with the same direction of effect. In some cases, they were SNPs in LD with those original SNPs, and we're seeing kind of a confirmatory signal replication, but not the exact model replication. The GWAS Catalog is an important and useful public database that we've incorporated into Loki, which is the knowledge base that biofilter draws its information. Other biofilter feature directions are we're currently integrating more public databases. We're really focused right now on the regulatory and non-coding regions. When we started biofilter, we were very GENIX focused, and we recognize that that's a huge limitation because we expect a lot of the interactions to occur between coding and non-coding regions. And so we're expanding that currently. We're also developing additional filter and model construction strategies based on specific hypotheses. So the one I mentioned based on the GWAS catalog, there are others that you can imagine kind of linking together coding and non-coding regions and setting up ways to filter and build models based on those. And we're also starting to work on a user interface for ease of use. It's currently strictly a command line application. We have a very extensive manual to go along with it, and so there are a number of users around the world, but we would like to make it easier for the public to use. And to wrap up, I just want to acknowledge the members of my lab and my collaborators. As I said, the HDL project was part of the eMERGE network, and there are far too many collaborators to list, but they were instrumental in pulling this data together. And then the MS project was part of the IMSGC. Again, far too many people to list, but I want to recognize their collaboration and allowing us to work with those data. And so I'll put up my last slide, which has my email address if there are follow-up questions. It also has my web address. The biofilter and Loki software is available for download from that website, along with the manual and some examples. And so I'll wrap up there, and hopefully there's time for questions.
There is indeed time for questions. So I don't see any on the, or at least I can't see any on the chat. Are there questions from anybody plugged in locally?
Marylyn, this is Lucia. Thank you for a very interesting talk. So one of the things that we've been thinking about with the GWAS catalog is just the limitations of the way that we currently assign traits, which is essentially, we try and semi-structure it, so we reuse terms whenever possible, but with some of the work that Helen and others are doing with ontologies, how would your sort of framework be able to take advantage of if we were able to map GWAS catalog terms to more defined ontologies and perhaps go beyond just a specific trait? Does that sort of factor into your pathway analysis?
Yeah, Lucia, that's a great question. I actually think that would make some of our work even easier. So I look forward to seeing that even more. So one of the things that I worry a lot about is that our definition of traits is too narrow and probably incorrect in a lot of cases. And so if we could use an ontology that would allow what individuals cite or study papers called a particular trait, kind of pull them together, it would allow us to bring together a lot of potentially similar traits, but they might not have called it the exact same thing. And for us, we would want to do that. So for example, if we were studying type 2 diabetes, we probably wouldn't only want to look at the original SNPs associated with type 2 diabetes and build the pathway models from there. We'd probably want perhaps all metabolic traits because genes that are associated with other metabolic syndromes may also be related to type 2 diabetes. And so I can imagine, using some of those ontology terms kind of as the branch point to pull our list of SNPs to start from to then build out these pathway models as opposed to looking at only one trait. So I think that would be fantastic. We want to be able to narrow our search space, but to still cast as wide a net as possible so that we don't miss too much. We don't want to narrow our focus to only look for things that we've seen before, but we need some prior knowledge to build from because there's just too much to look at across the genome.
So Marylyn, this is Helen. I kind of have a follow-up question. So when you were talking about how you were filtering your choices of interactions and the biological evidence for the MS analysis, you said that you required 4 biological databases to be substantiating the interaction. And I was wondering if there's some kind of pattern that you have databases which substantiate interactions more frequently than others, and if there's a biological reason for that, or if it's just coverage.
Yeah, that's a great question. So actually, it was in the MS study we didn't require 4 sources. It was in the lipid study. That's okay. I went through a lot of information pretty fast. So originally, the grad student that was working on that said he wanted to be more stringent and look at fewer models, and so he used 4 sources. We've just started looking into the relationship between the sources that we've captured. So, one question that I have is, are they highly correlated with respect to what original publication or PubMed ID they're pulling the information from? Because if they are, then it's not more substantial evidence. It's just that they happen to have both hit the same paper. Whereas if there's evidence of that interaction, let's say in the protein-protein interaction database from a yeast study, and then in KEGG based on some pathway information from some other in vitro assays, that would be kind of more substantial evidence as opposed to just that the multiple databases hit the same PubMed ID. So we've been looking at kind of building up that correlation structure, how the databases are linked together, and then also trying to figure out if there is kind of a number of sources that you would see biological evidence that would be more often than you would expect by chance. And so we've actually been doing some permutations of the Loki database to see, how often by chance do you see the same gene-gene combination in 7 or more databases. It's become much more complicated than we originally thought because it's not just the relationships between the genes, but it also seems to come into play the number of groups those genes participate in. So there are some genes that are represented in a dozen or more pathways, and so those can complicate this permutation idea a bit. But we've been investigating that in a lot of detail recently because people would like to be able, so we've been calling that the number of sources that models are observed in an implication index. And people have been asking, could I use the implication index as a Bayesian prior? Or can we put a p-value on the implication index to indicate that we're observing an interaction more than you would expect by chance? And as I expected, as we go down this rabbit hole, it's been much more complicated than I thought, but we're kind of in the thick of that right now.
Okay. That'll be a really interesting set of analyses.
Thank you.